Hello everyone, this is Rohan Mishram, and today's lecture we would devote ourselves to discuss as to what is PDB flat file format. In my previous video, we have discussed some aspects of structural biology, like as to how do the structural data is obtained using typically using X-ray crystallography, and I have also shown you a kind of demonstration in which you can judge what are the files and how you can obtain a good PDB file from public repositories like PDB. Okay, we have discussed facts like what is resolution and what are R value. Okay, so in today's lecture, as I have already mentioned, we would talk about as to how the structural data is arranged in a typical PDB file. Okay, so this is very important for you to understand, because it might happen in your subsequent career that you might want to change something in your PDB file, and in that case it would become very handy for you if you know as to what things in your PDB file are needed to be changed, and where exactly to locate that data and how to make those changes, so as to make that protein model as you want to work with. Okay, so keeping this in mind, it is very important that you should know as to how the data, or you can say the structural data, is arranged in a typical PDB file as such. Okay, so let us start out with this. For our today's demonstration, we would need some PDB file, some structure file. Okay, so let me first show you as to how do you download the uh, PDB, or you can say any protein model from protein data bank. Okay, for that all you need to do is to just visit PDB website. Okay, try googling it. Okay, then this is the site that you are interested in: Research Collaboratory for Structural Bioinformatics, RCSB PDB homepage. So once you reach to the home page, let us download our model molecule that we are going to use for most of our practicals, or for most of our demonstration today, as well as in our subsequent lectures as such. Okay, so each and every structural model that is deposited in PDB, they are provided with a specific unique identifier, and usually the PDB identifier are in form of 4 characters. Okay, so I know what is the unique PDB identifier of our own model: 1HEW. Okay, so 1HEW is the identifier of the protein molecule that we wish to work with. So you can search using that unique identifier, and once you search, you would get some hits over here. Okay, so this is what we are interested in. So click on this 1HEW, you would be taken to a separate page for that particular model. Then we want to download this model. So for downloading this model, you can use this download files button. Okay, and there are lot of other formats that are being offered to you, in which you can download the PDB or the structural information of your model. So we'll say, for example, you can download the sequence of that protein molecule in FASTA format. You can download the PDB file in text format. You can download that molecule in PDB format that is having this extension .gz. Okay, you can also download this structural data in mmcif format or xml format. Okay, there are a lot of options. Okay, that would completely depend on what you want to do with your pdb file or your structure file as such. Okay, so for our purpose we would require our structural model of our protein molecule in text file or in text format. So for that you would need to use this particular option. Okay, download the molecule in pdb format. Okay, just click on it. It will ask you where to save it. Okay, then you can say okay, and it would be downloaded in your download folder. Okay, so I have already downloaded it, and I have placed it on my desktop for the sake of simplicity. Okay, so this is what we need, right? So usually for structural visualization there are many options which are available. There are many computer software that you can use. Okay, you can use we'll say for example pymol, you can use rasmol, you can even use vmd. But for our demonstration purpose, most of the time we would be dealing stuff with spdbv. Okay, so you can search for spdbv. Okay, go to this download button or download page of Swiss PDB Viewer. You would be taken to their website from where you can download the you can you need to kind of agree on these terms and license. And since I expect that most of you must be working with Windows, so you need to download your software from this Windows option. Okay, and there would be a zip file. If you save this zip file, it would be saved in your download folder. Okay, and I have already done it. So what you can do is that you would get something zip file like this. Wherever you have saved your zip file, all you have to do is to extract this the information in this archive to say some in your C drive. Okay, I have extracted it in my C drive. Okay, you would get a folder like this wherever you would extract it, and in this folder you can run your spdbv through this application icon. Okay, so what you can do is that you can right click and you can send it to desktop, so that you in this way you would create a kind of shortcut to this particular application on desktop. If you use this send it to desktop, that is you would create a shortcut. Okay, so if you do this you would get an icon on your desktop something like this, and we would be calling this spdbv from our desktop every time when we work. Okay, so this is how you install your spdbv. Okay, so we would use this spdbv in a more extensive way in our upcoming practicals. But the main objective of today's practical is not visualization, but to understand how data is arranged in your PDB file. Okay, so well, say for example, you can just visualize any of your molecule just by dragging and dropping it into your SPDBV, and it generates a kind of beautiful three-dimensional model. But this is not what we want to do today. Okay, we would come to visualization stuff in our subsequent 2 or 3 or remaining lectures. We would be doing a lot of stuff on visualization, but today's objective is to kind of understand as to how this data is arranged in PDB file, how the data from PDB file is interpreted by this software to generate such three-dimensional model. That is what our major focus of discussion is for today, at least for time being. Okay, so Well, now if you recall that we have downloaded our protein structural information in text format. Okay, so here is what you can download from here. We have downloaded it, and we have downloaded that structural information in text format. Okay, so since it is saved in text format, you would also require a text editor for visualizing the text information. Okay, so there are many text editor which are available. Okay, so well, say for example, for my purpose, I am going to use a text editor called as TextPad. Okay, but I'm not sure if this TextPad is free software. Okay, so what you can do is that you can use one of the other text editor like Notepad plus plus. Okay, so you can download this Notepad plus plus from its website. Okay, and once you download, you can you know you can I will show you how it would look like. Okay, so the installer would look something like this. All you have to do is to double click on it, follow all the instruction, and you would install your text editor as well. We are going to need this text editor. Okay, so one way round to see what is provided in your PDB file is to directly visualize it in any protein structure visualizer. Well, so for example, in this case, we are visualizing your protein molecule in SPDBV, and the other way around is that you can see whatever information that is present in your PDB file, you can visualize it using any text editor like Notepad++. So you can visualize your textual information. It is the same file that we have loaded into different software. Over here it gives you three-dimensional representation, and over here it tells you as to how the structural information is arranged. Okay, so now why do we need specialized software for visualization of, you can say, the textual data that is arranged? Okay, now you can ask yourself a question like, I do have a default text editor in my Windows system, that is I can see it in Notepad also. Then why do we need a separate text editor? Okay, now to answer this, let us try to open it using your Notepad and see how things would appear, right? So this is how things would appear when you try to open that PDB file in your TextPad, and it looks quite chaotic. Okay, so you won't be able to understand what is exactly written in your PDB file. But if you open it in, say, some advanced text editor like Notepad++ or TextPad, the same file would appear something like this. So now it appears to be quite organized, and you can make out of things like what exactly is written. Okay, so that is why we require uh, some text editor. So programming people like bioinformatician or software engineers use these advanced text editors like Notepad or TextPad to write computer programs, the codes of their computer programs. I must be doing some. I was writing some codes. This is how codes of your computer program are written. So your text editor are different from your you can say word processor. Okay, word there are programs like word processor that handle text information like Microsoft Word. Okay, so Microsoft Word is a word processor wherein you can do lot of things like shading and changing the font and everything. Okay, so if you want to do that thing, you can open you can use word processor. But if you want to use just text manipulation, then text editing is usually done in text editors like Notepad++. Okay, so this is how your text editor differ from word processor. Okay, in text editor there is no option of doing italics and different fonts and highlighting and everything, right? So those are not meant to be done using text editor, while word processor you can do all the formatting and everything. Okay, so what my point is that we would require some text editor for today's purpose. Okay, good. So I hope now you know as to how do you open the same PDB file in three dimension to visualize it in three dimensional, or you can say uh, to visualize the three-dimensional structure of the protein. And in order to see what is written in that PDB file, you can use any text editor, like you can use this Notepad. Most of the time I would be using this TextPad. I'm comfortable with TextPad. Okay. I'm sorry. So well, now let us focus on as to how the structural data is arranged, or in other words we would be discussing what is PDB flat file format. Okay, so well, say for example, let us view the line numbers, right? So for the sake of simplicity, you can say that the PDB file that we are dealing with, or we would be dealing with for most of the time in our practical session, it belongs to an enzyme called as lysozyme. Okay, and this lysozyme was derived from the white portion of the hen egg. Okay, so this lysozyme, which is derived from the white portion of hen egg, is crystallized and that is deposited in PDB, and that is what you have downloaded. Okay, and this is the enzyme that is said to be that is said to be crystallized in combination with its inhibitor called as trinag. Okay, that correspond to tri and acetyl chitrose at 1.75 angstrom resolution. Okay, so this is the model that we are going to use for our next subsequent practicals. Okay, so now let us try to understand as to how structural data is arranged in this PDB file. Okay, so for the sake of study, you can say that any PDB file is divided into two major sections. Okay, so and each and every PDB file, uh, each and every PDB, the data, the structural data is arranged in lines as such. Okay, and each line begins with a specific keyword. So we'll say for example, your PDB file contains various keywords that are arranged on, say, separate lines or rows, you can say. Okay, so how many lines are there in this particular PDB file? Well, let us scroll down. This column indicates the line count. So this is first line, second line, third line, fourth line, and fifth line. Okay, so in this way, if we scroll down, you can see that this PDB file consists of data that is extended over 1627 lines. Okay, and this 1627 lines is read by your structure visualizer the moment you drag and drop it, and it would show you this beautiful model. Okay, so it is the how fast these computers are, so that in just blink of second it has read all these 1627 line and showed you this beautiful model. Okay, so now our objective is to kind of explore what are the different keywords, or what are the different lines that are present in your PDB file, and what do they signify. Okay, first we could try to understand that, and then we would try to kind of correlate as to what data is read from this PDB file to generate that three-dimensional model. That is what we will do today. Okay, so now to begin with, for the sake of simplicity, as I have already mentioned, any PDB file, can be divided into two major portions. Okay, the first portion is called as the annotation portion of your PDB file, and the majority of your PDB file is represented in coordinate section. Okay, so for time being, if you understand that you can divide your PDB file in two sections: annotation section and the coordinate section, wherein the structural information is actually provided. In annotation section, some information is provided for the for the researcher to read and to understand as to what that protein structure is all about. Okay, so the annotation section would provide you some information, some background of that protein molecule, while the coordinate section will provide you the structural data that is used by these visualizer in order to generate the 3D model. Okay, so now let us focus on as to what information is provided in such annotation section. Okay, usually, annotation sections are present at the very beginning of your PDB file. Well, say for example, your annotation section, which begins with the first line, uh, and it has got a lot of lines which begins with different keyword. We would come to these different keywords in a while. Okay, so your annotation section start out from line number 1 to line number this much, 426. Okay, so you can just guess the length of your annotation section from over here. So from first line to line number 426, whatever data is present, you can consider it as annotation section of your PDB. And whatever lines that are present in PDB, in your PDB file, that begins with the keyword atom or heteroatom and connect lines, those represent your coordinate information. Okay, so now you can say that of the 1627 line in your PDB, only 426 lines are devoted in annotation section, and remaining lines in your PDB would correspond to your coordinate information. Okay, we will come to what exactly is coordinate information in a while. Okay, so 1/4 of your PDB file is devoted to annotation section, while 3/4 of your PDB file is devoted to your coordinate information. Okay, so now let us try to understand what are these different keywords that are present in your PDB file and what do they signify. Okay, so the very first and usual keyword that is present in any PDB file is called as the header line, or the line that begins with the keyword header is called as the header line. Header line would give you information like as to when that when that molecule was deposited in your protein data bank, what is its uh, unique identifier. Okay, the lines that begins with title will give you an idea as to what is the title that is present in PDB, by what title this particular model was deposited in PDB. Okay, do you remember we have downloaded this RC, this model from PDB, and it has got title like refinement of an enzyme complex with inhibitor bound at partial occupancy, blah blah blah. The same title is present over here, refinement of an enzyme complex with inhibitor bound to partial and everything. Okay, so this gives you a kind of the title lines would give you information as to what is the title of this PDB file. Okay, then you have got this compound information wherein it will give you idea as to how many chains are there in your PDB, what is the EC or enzyme commission number for that particular enzyme. Okay, then there are source lines which gives you information about as to what is the source from which this particular protein was isolated and crystallized, right? Then there is, if the line begins with this keyword is used for telling the user as to what was the experimental method that was used to obtain the structural data of this protein. So this particular protein was solved using the technique called as X-ray diffraction. There are other techniques like NMR by which structure of protein can be determined, but this particular protein was solved using using experimental technique called as X-ray diffraction. So author line gives you as to how many authors were involved in that study. Rev dat is the this particular set of lines, or this particular set of keywords would give you an idea as to how many times this particular entry was revised. The very first time it was deposited on 31st January 94, then it was revised on 1st April 2003, then on 24th February 2009, and late, latest it was revised in 2017 on 29th of November. And what portion of this PDB file was was revised is shown over here. Okay, then this lines that begin with J, R and L will give you an idea where that model is published. Okay, so the lines that begin with journal and then author gives you the idea as to what is the list of authors, that uh, list of authors where the model was published. Okay, what is the title of that paper, what is the in what journal it was published, what is the volume information, what is the page information. Okay, and you can have other journal information like what is the ISSN number of this journal in which the model was published, what is the digital object identifier of that paper. Okay, so this such type of information, the citation details is provided in this JRNL line. Okay, then you have got a very big chunk of this remark line. Okay, so I won't go in more details of what remarks might be there. It is completely dependent on what remarks that author wants to give. Okay, so remark line are usually given to express or to share what was the experimental observations. Okay, like how they have calculated the R value, R free value, and we'll say for example what was the size of the crystal, and what was the type of the crystal, so on and so forth. Okay, we won't go in more details of this remark, right? So if you want to know as to how many type of keywords a particular PDB entry might have, you can search in PDB, check out their documentation page, and there you can figure out as to how many keywords and keywords might be there in your PDB file and what would that keyword mean. For time being, I am only showing you what are the keywords that are present in our particular PDB file and what do they mean, right? So, well, say for example, after remarks, you have got this sec res lines that begin with keyword sequence residue. Sec res stand for sequence residue, right? And over here, you have been provided with what are different amino acid residues that are present in your protein. Okay, along with the amino acid residue, this het line would tell you as to what are the hetero groups that are present in your protein. Okay, so hetero group are usually the non-protein component that are present in your file. Okay, this non-protein component can be in form of prosthetic groups like PLP or ADP, ATP, and so on and so forth, right? NAD, NADP. These are all prosthetic groups. The heteroatom can be, or you can say the non-protein component can be in form of metal ions, like most of the protein requires some metal ions for their activity. Well, for example, you can find some protein that are co-crystallized with magnesium, some manganese or calcium, so on and so forth. So the structural data of all of the entities that are present in your proteins that are non-protein in nature are provided in this heteroatoms as such. We would come to this heteroatom description in more detail. Okay, then you have secondary structure information. Okay, as to how many helices are there in your protein, how many beta sheets are there, how many disulfide linkages are there. Okay, so on and so forth. Okay, so there are different. The point is that in your annotation section there can be variety of keyword containing different information. Okay, so this is all about your annotation section. Okay, and let us now begin with another section of your protein file, and this is very heart of your PDB file. Okay, so we call it as coordinate information. Okay, so all the lines that begins with atom keyword, okay, atom, eventually the line that begins with heteroatom and the connect keyword. Well, all of this data is included in your coordinate information. Okay, or we can call it as coordinate lines. Okay, and majority of your PDB file, or you can say the bulk of your PDB file, is occupied by these coordinate lines. Okay, so now let us try to understand as to what exactly these coordinate lines are. Okay, what do these coordinate lines signify? Okay, so these coordinate lines are quite very easy to understand. So it is this it is the data from this coordinate it is the data from this coordinate line that are read by your visualization software to generate the model. Okay, and let me first tell you as to what are the coordinate lines that are present in your PDB file. What do these lines signify? What do the data in this line signify? Okay, so okay, okay. So as you can see over here, the coordinate information always begins with this with the line that starts with the keyword atom, ATOM. Okay, So this keyword atom, well, this line you can say has got how many columns? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11. Okay, it has got 11 columns, and structural information is usually provided in these 11 columns. Okay, so what you can count over here, that the lines that are continuous throughout this atom line, okay, or coordinate, this is this indicate that this is the first atom in your PDB file. This line correspond to the second atom in your PDB file, third atom in your PDB file, fourth atom in your PDB file, so on and so forth. In this way, if you scroll down, how many atoms are there in your PDB file? You can just scroll down that in your protein molecule there are 1001 atoms in your protein molecule. Okay, and this heteroatom would correspond to non-protein component. We would come to this in a while. So one it is now, I think it is clear that uh, your your line information is continuous throughout the PDB file. Okay, the atom information that correspond to the structural information of first atom, second atom, third atom, it is indicated over here as to how many atoms are there in your protein molecule. You can get that from over here. Okay, so after this atom line, you can say after this atom line there is a column that signify these letters N, C, A, C, C, B, C, G, N, N, Z, C, E, and N, Z. So this particular column corresponds to what are the atoms that are present in your protein molecule. Okay, well, say for example, well, let us focus on the very first residue. Okay, so well, I will again show you as to how can you load this. This is the file that we have downloaded, right? You can kind of drag it into your SPDBV software, and you would load the same molecule, and this is the same molecule that we have opened it in any text editor, right? So when you open it in SPDBV, now let us see as to how many amino acid residues are sorry, are there in your PDB file. For that you would require this control panel. Okay, let me arrange these things properly. So in your control panel, your amino acid residues that are present in your protein are listed in this particular control panel. So at first position you have got lysine, at second position the amino acid residue valine is present, at third position phenylalanine is present, so on and so forth. Well, for time being, let us focus. I am showing the ribbon representation. For time being, let us focus only on the first amino acid residue, that is lysine. Okay, so here it is located in your protein. Okay, so this is the first amino acid residue in your protein. Now I am letting this I'm showing you in your ribbon representation as to just as to give you a kind of idea as to where this protein where this amino acid residue is located. Okay, so it is present on this corner of your protein molecule. Okay, so now you get an overall idea as to where that lysine 1 is located in your protein. Okay, so let me hide this ribbon and let us focus only on our lysine residue by labeling it. Okay, so here we have labeled our lysine group. So let us zoom on this lysine molecule. Okay, so in this way what you have done is that you have labeled this much portion from, sorry, you have labeled this much portion of your PDB file. Okay, so what you are visualizing over here is the structural data that is represented on the line number 427 till line number 435. Okay, and when you click on this label button, you have labeled this lysine. Okay, okay. So now, in order to understand in more details, we have just labeled the amino acid residue and the number. For the sake of simplicity, you can display the labels in atom name so that you can visualize what are different items that are present. Well, now let us come back to the text information. So this is how the structural data is arranged. You know, conventionally, when you represent any protein molecule, it is represented from its N terminus to C terminus. So this N correspond to the N terminus of your protein, and the last atom of your protein is the terminal oxygen, or what we call it as OXT. Okay, so in this way you represent your protein molecule from N terminus to C terminus. Okay, so usually the structural information of any protein begins with the N terminal residue, that is the first nitrogen from the first lysine group. Okay, so that is what this N represent. Okay, so in this way this nitrogen is your in 3 dimension it is represented over here. Then you have got C alpha carbon atom that is attached to this nitrogen. Then you have got this carbonyl carbon atom that is attached to this C alpha carbon atom. This is the carbonyl carbon. To this carbonyl carbon you have attached the oxygen, which is this. Okay, to your C alpha carbon atom you have got this C beta carbon atom, you have got this C gamma carbon atom, which is represented over here. Okay, then you have got C delta carbon atom, which is represented over here at 7th position. Then you have got C epsilon carbon atom. And you know, at the end of any lysine molecule there is a nitrogen atom or amino group that is present over here. Okay, so actually what you are visualizing is the information that is present from line number 1 to line number this much. This is the information that you are visualizing over here. Okay, so after these atoms that are present in your first lysine group that you are visualizing, the next column that is present is for the 3-letter code for amino acid. So this is lysine, the 3-letter code of lysine is LYS. Okay, the subsequent column will tell you as to in what chain this particular amino acid residue is present. So it is present in chain A. Proteins can be multimeric in nature as well. Okay, so there can be different chains if it is multimeric, chain A, chain B, right? So in our case, this lysine is from chain A, that is what it says. Okay, and this particular column would tell as to this is the first amino acid residue. The data from this line to this line belongs to the first amino acid residue, that is lysine. Next subsequent amino acid residue, the data for next subsequent amino acid residue is present from line number 436 to line number 442, that belongs to valine, which is present at the second location. Do you see over here? Valine is present at second location and its corresponding data is represented just below the first amino acid residue. Okay, so this particular column will tell you as to what is the number of that amino acid residue in its primary structure. The lysine at the first position, valine at the second position, you have got phenylalanine at the third position, so on and so forth. Okay, so what is the number of that amino acid residue is represented in this particular column. Okay, so next three columns correspond, the next three columns correspond to the actual coordinate of your PDB file. This is very important. So these are this for this particular nitrogen atom. In order to represent this particular nitrogen atom, the structural data of this nitrogen atom is represented in Cartesian coordinates. Okay, and the in order to represent anything in your Cartesian coordinates, you would have to provide the three coordinates of each and every atom. So this is the x coordinate of your nitrogen, this is the y coordinate of your nitrogen, and it is the z coordinate of your nitrogen. Okay, so these are x, y, and z coordinates of this nitrogen in your PDB file. Okay, to make it more clear, I would just represent it in, well, say for example, let me just tell you. Okay, so what I have said is that the atoms in your PDB file are represented in 3D coordinates, or you can say three-dimensional coordinates. You have got x coordinate, y coordinate, and z coordinate. So what do this coordinate mean is simple. Okay, so let me see if I can let us copy this thing. Okay, copy and paste it over. So here are the x coordinate, y coordinate, and z coordinate. Okay, so and these coordinate are for your first nitrogen atom. So what do these x coordinate, y coordinate, and z coordinate actually mean? Okay, so as I have already mentioned you, each and every atom in your PDB file is represented in Cartesian coordinate. So this is your x-axis, this is your y-axis, and this is your z-axis. Okay, the z-axis usually comes out of plane, but since we are using two-dimensional picture, I cannot show you how this comes out of the plane for time being. Let us consider that this is your z-axis. Okay, and we are talking about this nitrogen. On x-axis there must be certain scale. This is 1 2 3 4 5. On y-axis similarly you have got the scale. Okay, 1 2, say for example 9 10, right? And z-axis also you might have certain scale, 1 2 10 11, so on and so forth. Okay, so these are the three axes of your coordinate system, or what we call it as the Cartesian coordinate system, which has got origin with 0. Okay, we call it as origin. So what do these coordinate represent is the location of your nitrogen atom. So your nitrogen atom is located on x-axis with a distance of 3. So over here this is your x coordinate. On y-axis it is located at the 9th position, which is over here. You can locate it as something like this. Okay, so this is the 9th position, this is the third position, and on z-axis it is located on 10th position. That is something like this. So this is what the computer program does. From the origin of your coordinate system, it reads out each and every line. Each and every line has got x, y, and z coordinate. So if you're dealing with the first nitrogen, okay, it would read out its x coordinate, which is y coordinate, which is present over here, how far it is located from the Z coordinate which is present over here, and it would place a blue dot over there which would correspond to your nitrogen. So this is how the data from your PDB file would be read. Okay, so each and every line in your PDB file would start out with an atom, and each and every atom will have the 3D coordinate. These first column corresponds to the x coordinate, the second column corresponds to the y coordinate, and the third column corresponds to the z coordinate. And these coordinate are in reference to, say, origin of your Cartesian system. Okay, so for the sake of simplicity, what you can imagine now is that the visualization software like the SPDBV that we have already used, what it will do, it will read this information line by line. It would start out with the first atom, that is nitrogen. It would read its 3 coordinate, its x coordinate, its y coordinate, its z coordinate, and appropriately place a blue dot over here as per the data that is present in these 3 coordinates. Okay, so in this way it would read the first line that correspond to nitrogen, read its 3 coordinate information, x coordinate, y coordinate, and z coordinate, and according to this coordinate place a blue dot over here. Then move on to the next line that correspond to C alpha carbon atom, read its x coordinate, y coordinate, and z coordinate, and place a white dot over here, and then join the blue dot to the white dot. So in this way it would iteratively read all of the information that is present or that starts out with atom line, and accordingly go on placing this red dot, white dot, or blue dot depending on what kind of atom that is present over here. And in this way, in your 3 dimension, you would generate this beautiful model. Okay, so iteratively it would read out all of the atom lines that start out with this, and when it encounters this termination line, that would indicate that the structural information of protein gets over here. The line that begins with the keyword ter, that correspond to termination, we call it as the termination line. This termination line indicates that this is the line where your protein information is finished, and the subsequent line would be devoted for heteroatom or non-protein component of your protein. Okay, so in this way the structural information is provided in the form of the 3 coordinates for each and every atom that is present in your protein. Okay, so these 3 lines, or you can say these 3 columns, correspond to the x coordinate, y coordinate, and z coordinate of your protein molecule. Okay, so the protein structural information begins with the lines that has got keyword atom and ends with the line that has keyword ter or termination line. So this is the take home message. Okay, so these are the 3 coordinate, x coordinate, y coordinate, and z coordinate for each and every atom in your protein. So besides these 3 coordinate, there are 2 more columns that are present in your PDB file, and these are also very important for you to learn. The next column that is present after the coordinate column is called as the occupancy. This column represent your occupancy of each and every atom in your protein molecule. Okay, and the column which is subsequent to occupancy is called as the B factor column. So this column correspond to the B factor of each and every atom in your PDB file. Okay, so now let us start discussing as to what exactly this occupancy, what do it signify, and what are B factor and what do they signify. Okay, So for that I think we would need our protein PowerPoint presentation. So this is what it is. In a nutshell, this is what we have discussed. Okay, so this is the atom number. This is the these are the name of the atoms. This is C alpha carbon atom, C beta carbon atom, so on and so forth. This column correspond to the residue name. Okay, in 3-letter amino acid abbreviation. This is the residue number. This is the x coordinate. This is the y coordinate, and this is the z coordinate. Okay, and we have kind of correlated this textual information to the three-dimensional information. This is how your computer program would interpret these lines. Okay, this particular line is for this nitrogen. This particular line is for this C alpha carbon atom. This particular third line is for this carbonyl carbon atom. Fourth line is for this carbonyl oxygen atom. Fifth line is for this C beta carbon atom, and subsequently uh, till you reach the terminal atom of your side chain. So of these lines, okay, this N, C alpha, C and O, N, C alpha, C and O are called as the backbone atoms, and whatever is present subsequent to C alpha carbon atoms would correspond to your side chain carbon atoms. So this much structural information from line number 5 to line number 9 of lysine first is devoted to your side chain, and from first line till the oxygen, this much information is for your backbone atom or main chain atom. As you know that the amino acid residue has got two components, the backbone component and the side chain component. Okay, now as I have mentioned you that there are two more columns called as the occupancy column and the B factor column. Okay, so B factor is also called as sometimes temperature factor. Okay, so let us start now discussing as to what are this B factor or what is called as the temperature factor. Okay, so this B factor, you know, however, you can see that this protein molecule seems to be static, right? In this model, it seems to be static, but at physiological condition, when the protein is present in cells, it is always involved under some dynamics. Okay, it entails dynamics. That is, the atoms are not static in protein, but they show certain movement when it is present in cells. Okay, so the atoms in the proteins are always in motions or they are in vibration to you can say varying degree depending where they are present. Okay, so usually the atoms that are present in the core of your protein usually do not move much. Okay, why? Because the core is usually stabilized, while the atoms that are present on the surface of your protein, they show a bit of movement. The side chains that are exposed to solvent, they usually show movement. Okay, so now depending on as to where your atom is located in your protein molecule, they might show some varying degree of freedom, or you can say the varying degree of motion. Usually the atoms that are present in the core of your protein, they have got less degree of motion, while the atoms that are present on the surface of your protein, they show higher degree of motion. Okay, and this vibration or the motion of the atoms in your protein molecule are accounted by something called as the B factor or temperature factor. Okay, so the temperature factor is usually, we are talking about this, the values that are present in your temperature factor would give you an idea about the motion of that motion of that atom in the protein molecule. Okay, so this value would indicate whether this particular atom, this particular nitrogen atom is moving well, or it is vibrating, or whether it is very stable. Okay, and what is the scale of these vibration then? Let us discuss that first. Okay, if the value in your B factor is under 10, that would indicate that the atom is not moving much. Okay, and if the value is greater than 50, that would indicate the atom is moving so fast that it can be barely seen in electron density map. Okay, it is very difficult for that atom to be located. Okay, so if the atom is having the B factor value under 10, that means it is quite stable in your protein molecule. While if the value of B factor is greater than 50, that would eventually mean that it has got inherent flexibility in it. Okay, so this is often the case for atoms. I mean, uh, the atoms which has got the B factor higher than 50 are usually the atoms that are present on the surface of the protein. Okay, and usually these are the side chains where side chains of amino acid residues that are longer in length. We'll say, for example, the arginine group or the lysine group, where the side chains can wag in the surrounding water. Okay, they can move in the surrounding water. Well, such side chains has got higher B factor. Okay, so now you can kind of visualize in your protein molecule as to what are the side chains that have got higher P factor. You can, you know, visualize it. I will show you. Okay, well, now you can color your protein molecule by B factor like this. Okay, so now when you color your protein molecule by B factor, it would color it as per the visible spectrum. That is, the atoms having lowest B factor would be colored in the shade of blue, while the atoms which are having highest B factors would be colored in the shade of red. Okay, so now as you can see, the atoms that are present in the core of the protein are, you know, in the shade of blue, while the atoms that are present on the surface of your protein are moving towards the shade of red. Okay, and the same is reflected over here in your control panel. Okay, so now this arginine 128 appears to be having one of the highest B factor. Okay, well, now let us try to locate where exactly this arginine 128 is located in your PDB file. Okay, it is one of the last amino acid residue 128, and you can see the data for arginine 128. The backbone atoms has got the highest B factor value 85 and 85, right? Do you see all of the atoms that are from side chain are having the B factors above 50. The highest B factor of the atoms is for this nitrogen atom from the guanidino group 85, 83, right? And hence they are colored in the shade of red. Okay, so it can move quite fast. Well, since it is exposed to the solvent, well, this particular side chain is long and it can wag around and interact with the surrounding water molecules. Okay, so therefore it has got higher B factor value. Okay, so this is all about your B factor. So higher B factor would mean that the atoms of this particular atoms in your PDB file are involved in motion, and the degree of motion can be inferred by coloring it with B vector. Okay, so that is all about your B vector. Okay, let us now talk about occupancy, or you can say multiple confirmation. Okay, so in order to understand what occupancy is, well, let us revise what we have discussed in our last lecture. So I have said that the macromolecules like proteins, when they are crystallized, what happens is that the individual molecules in your the individual molecules, well, they get packed into symmetrical arrangement. Well, say for example, if this is the protein molecule, and when they crystallize, they are arranged in symmetrical fashion into that forms a kind of crystal. Okay, and whatever molecule that you visualize over here, that you see over here, is an average molecule that is present in your entire crystal. So when you crystallize, each and individual protein molecule would arrange itself, or you can say the crystal is nothing but symmetrical arrangement of the number of protein molecules. And what you see in the model is a normalized coordinates of each and every protein, each and every atoms in your crystal. Okay, so now here comes some more interesting fact. So, as I have said, the macromolecular crystals are composed of many individual molecules that are packed into symmetrical arrangement. So now, in some crystals, there might be slight difference. Okay, so there can be differences like side chain on the surface. As I have already mentioned, it can wag back and forth between several conformation. Okay, so well, say for example, in case of this particular molecule, the side chain can be like this, while in this particular molecule, the side chain can orient in some different way, the same side chain can orient in different way, right? So in the same crystal, there can be different arrangement of atoms in your protein molecule. Okay, and these differences in your protein, the differences of arrangement of atom in your protein molecule in a same crystal is usually represented in the form of your occupancies. Okay, another way around is that the substrate may bind in two orientations in active site. If this is an enzyme, then when a substrate bind, it can, in half of the molecules in your crystal, it can take this orientation, in half of the molecules it can take some different orientation, right? So the substrate or the inhibitor can be differentially oriented in each and every protein molecules in your crystal. Okay, it can also happen that metal ions may be bound to only few of the molecules in your crystal. So out of, we'll say for example, 1,000 molecules, or out of, we'll say for example, 100 protein molecule that are present in crystal, it is possible that the metal ion can be bound to only 50 molecules in your crystal, and 50 molecules can remain unbound from the metal ions, right? So these things can happen during the process of crystallization. So the term occupancy is a kind of estimate as to the amount of each conformation that is observed in the crystal. Okay, so in this way, if the occupancy is given a value of 1, that would indicate that the atom is found in all of the molecule in the same place in the crystal. Okay, so if the same set of atom is present at the same location in all of the protein molecule in your crystal, then the occupancy is given the value of 5, value of 1. Okay, if the atom is present only in half of the molecules in your crystal, the occupancy would be given to the value of 0.5. The value of occupancy 0.5 indicates that the atom is present in the same location only in half of the molecules in your crystal. That is what this occupancy mean. Okay, so for example, if a metal ion binds to only half of the molecule in crystal, the occupancy value would be 0.5. Okay, so this is how the occupancy might be represented in your electron density map. Okay, over here you can see that in half of the crystal, the side chain is oriented in this way, while in half of the crystal, the side chain might be oriented in this way. Okay, and in such a case, the occupancy for this particular atom in your PDB file would be 0.5. Okay, in second example, as I have shown you, this tyrosine side chain in half of the molecules in your crystal, it is oriented in this way, while in half of the molecules in your crystal, it is oriented in some another way. In such a way, well, say for example, in 75% of the time, if it is oriented in this way, the occupancy would be 0.75, so on and so forth, right? So the occupancy would indicate multiple conformation if the atom is involved with. Okay, so in this way, the occupancy of this particular atoms, this set of atom is 1. That would indicate, that would eventually indicate that all of these atoms has got the same position in all of the molecules that are present in your crystal. That is what it indicates. Okay, so this is how your data is arranged in your PDB flat file format. Okay, one more thing that you might have noted. Okay, I would again color it back to CPK. That again, let us focus on the first lysine. Okay, let me label it by the name of atom, and you can now see, as I have shown over here, that you have been provided with data of only heavy atoms. Okay, so this is the C alpha carbon atom, this is this is the carbonyl carbon atom, this is C alpha carbon atom, this is C beta carbon atom, C gamma carbon atom. Okay, but now you can, don't you wonder that only the information of the heavy atoms is provided over here, right? So there is no information of hydrogen atoms. Okay, so if you only represent the carbon atom, the valency would remain unsatisfied. Okay, so if this is the carbon atom, then it should be like CH2, it should be like CH2 over here. There should be 2 hydrogen atoms present over here in order to completely satisfy the valency of your molecule, or your amino acid residue. But no information regarding your hydrogen atoms is present over here. Okay, and Why is that? Why is that we don't get any information of hydrogen atoms in specifically if the protein is experimentally solved using the techniques like X-ray crystallography? You won't get any information of the hydrogen atoms that are attached to it. Why is that? Okay, the answer lies is that uh, you know hydrogen atoms has got only one electron in its outermost shell. Okay, and the remaining atoms has got multiple electrons in its outermost shell. Like carbon has got multiple electrons, nitrogen has got multiple electrons. More is the number of electrons in around those atoms, you can say that the electron density around that atom is very high. So the heavy atoms are those atoms besides the hydrogens that are present in your protein molecules. Okay, heavy atoms can be carbon, nitrogen, oxygen, sulfur. Those can be included in heavy atoms. So these heavy atoms has got higher electron densities as compared to hydrogen atoms. Why? Because hydrogen atom has got only single electron as compared to other heavy atoms. And when such heavy atoms are exposed to X-rays, due to higher electron densities, the X-rays would get deflected with higher magnitude due to higher electron density cloud around them. On the other hand, your hydrogen, which has got a single electron in its outermost shell, well, it might not diffract the X-rays in higher magnitude so that it can be detected on the detector. Okay, so since the electrons or the electron cloud is not detected from hydrogen, the spot of hydrogen atom won't appear on your detector. If the spot is not detected on your detector, there would be no intensity for hydrogen atom. If there is no spot and no intensity of hydrogen atom, you cannot obtain the electron density map of hydrogen atoms. No electron density map, that means hydrogen atoms cannot be resolved in this case. Okay, on the other hand, heavy atom has got higher electron density. They diffract the X-rays quite very well so that the spots for the corresponding atoms can be obtained on the detector. If you obtain the spots, if you can obtain the intensities, you can eventually obtain the electron density in your electron density map for each and every heavy atom. So this is the reason, my friends, that if the protein model is solved using X-ray crystallography, the electron densities of your hydrogen cannot be resolved, and hence the position of hydrogen atoms around the heavy atoms are not usually detected in X-ray crystallography. Okay, so you would have to assume that there are two hydrogen atoms present around this C beta carbon atom. There are two hydrogen atoms that are present around this C gamma carbon atoms, so on and so forth. So this is the reason why hydrogens are not present in your PDB file. Hydrogens are not detected in your PDB file. Okay, so this is all I have to discuss about your PDB flat file format, and I hope now you understand as to what data in your PDB file is read by this visualization software, and how these beautiful models are generated, and you can visualize it and you can analyze it. Okay, so this is all, folks, that I have got you so far your PDB format is concerned. In our next lecture, we would discuss more details of the visualization of your structural data. Okay, so this is it. Thank you for your kind attention, and I would see you in our next lecture.